hemoglobin, like oxygen, it combines with heme group, not with amino group, forming a stable compound, and the stability is 200 times greater than that of oxyhemoglobin. The stability of carboxyhemoglobin. This is carboxyhemoglobin. This is HbCO2 carbaminohemoglobin. The affinity of carbon monoxide with heme group is 200 times greater than that of oxygen. That's why when hemoglobin combines with carbon monoxide, it forms carboxyhemoglobin, which is a stable compound. Whereas carbaminohemoglobin, oxyhemoglobin are reversible compounds, can easily be break down. They can be break down very easily. But carboxyhemoglobin cannot be broken very easily because it's a stable compound. It is highly dangerous. Next one. In RBC, there is one enzyme what is called carbonic anhydrase. The same enzyme is also present in plasma, but high concentration of carbonic anhydrase enzyme present in RBC. Low concentration of low amount of carbonic anhydrase is present in plasma. So the concentration of carbonic anhydrase is more in RBC, less in plasma. This is another important. A multiple choice question may be given. The presence of carbonic anhydrase in the blood, less in plasma, more in RBC. More in RBC, less in plasma. Equally present both in plasma and RBC. Only present in RBC and absent in plasma. In such a way, 4 options will be given. So the enzyme carbonic anhydrase concentration is more in RBC and less in plasma. This anhydrase enzyme is responsible. It catalyzes the reaction, that is H2O plus CO2 combines to form H2CO3, carbonic anhydrase, that immediately breaks into H plus and HCO3 minus. H2O plus CO2 forming carbonic acid that dissociates into hydrogen plus and HCO3 minus ions. This reaction is catalyzed by an enzyme, what is called carbonic anhydrase. Important point. Whose concentration is high in RBC and less in plasma. In total, 70% of carbon dioxide is transported by the blood in the form of bicarbonate ions. So carbonic acid immediately dissociates into hydrogen and bicarbonate. Again, they combine to form hydrocarbonic acid. This reaction is catalyzed by an enzyme what is called carbonic anhydrase, whose concentration is high in RBC and low in plasma. Now, at tissues, CO2 is released by catabolic reactions, breakdown of food materials. Along with the CO2, H2O, energy is produced. By that, the temperature also increased in the cells due to cellular respiration. So high temperature, why it exists in the tissues because of catabolic activity. So CO2, H2O and energy is liberated, but temperature increases at the tissues. Now the CO2 and H2O combine to form H2CO3, dissociates into HCO3 minus and H plus. This reaction occurs both in plasma and also in RBC. This formation of carbonic acid and dissociation of carbonic acid into hydrogen and HCO3 ions is catalyzed by the enzyme carbonic anhydrase, which occurs both in RBC and also in plasma. That is another important point. You see, bicarbonate ions and hydrogen ions both reach the alveoli, where again they form into carbonic acid and dissociates into H2O plus CO2. CO2 goes out from alveoli. In pulmonary ventilation during expiration, the carbonic acid dissociates into water and carbon dioxide, yet alveoli and carbon dioxide is released out. This is the process of the transportation of bicarbonates. So, in three forms, the carbon dioxide released at tissues by cellular respiration. Number one, in dissolved state, about 77% of carbon dioxide is transported by the blood, and about 20 to 25% of carbon dioxide is transported by the blood in the form of carbaminohemoglobin. Carbaminohemoglobin is found by the combination of carbon dioxide and hemoglobin. Carboxyhemoglobin, a stable compound, is found by the union of carbon monoxide and hemoglobin. Carboxyhemoglobin, HbCO, carbaminohemoglobin, HbCO2, HbO2, oxyhemo. Right. For your understanding, today I will take 10 more minutes extra to complete this respiration chapter. Before that, I would like to draw two diagrams. By that, the transportation of carbon dioxide can easily be understood by all the students. It is not mentioned in Neet textbook. For your understanding, I will draw the diagram. Very easy. See here, at the tissues. This is tissue. Okay. In tissues, carbon dioxide is produced by catabolism. Okay. Now, this is blood, plasma. Okay. This is RBC. This is RBC plasma. Now, how the gases are exchanged between blood and tissues. First, carbon dioxide, how it is transported, how it is diffused to the blood. How much percentage of carbon dioxide is transported in dissolved state? 7%. So, CO2, dissolved state. Okay. How much? 7%. CO2 enter into RBC, where it is combined with the hemoglobin, forming carbaminohemoglobin. This is how much percent? 20 to 25%. Okay. In the third phase, CO2 enters into RBC, CO2 plus H2O, forming H2CO3, dissociates into H plus and HCO3 minus. Result calculated. So, bicarbonate and hydrogen ions are formed in RBC and also in plasma. CO2 plus H2O, H2CO3 dissociates into HCO3 minus plus H plus. This reaction also occurs in plasma and also inside the RBC, and this is catalyzed by the enzyme carbonic anhydrase. So, more amount of bicarbonates are produced in RBC than that of plasma. Okay. Now, the bicarbonate ions, they should be transported only through plasma, not through RBC. Not through RBC. You see, the hydrogen ions, which are produced by the breakdown of H2CO3, combined with the oxygen, combined with hemoglobin. The hydrogen ions are attached to hemoglobin. Here, HCO3. This HCO3 bicarbonates come out. And in reciprocation, chloride ions enter into the RBC to maintain ionic equilibrium. So all bicarbonate ions which are formed in RBC, they come out from RBC. To maintain equilibrium, chloride ions from plasma enter into RBC. Chloride ions plasma enter into RBC. This is what is called chloride shift. Chloride shift or Hamburger's phenomenon. Hamburger's phenomenon or chloride shift. So when carbon dioxide enters into RBC, combines with water, forming carbonic acid, dissociates into hydrogen ions and bicarbonate ions. And the bicarbonate ions come out from RBC, and in reverse, chloride ions enters into RBC. The exchange of the chloride and hydrogen bicarbonate ions across the RBC membrane is what is called chloride shift or Hamburger's phenomenon. Hamburger's phenomenon. The same reaction occurs here. So total bicarbonate ions are transported in the plasma up to alveoli. Up to alveoli. These are the changes occur at the tissues. Carbon dioxide. We say HbO2 at tissues. HbO2 dissociates into Hb plus O2. O2 is supplied to the tissues. The hydrogen ions combine with the Hb plus H, forming HHB, what is called reduced hemoglobin. You know, oxygen is transported to tissues. HbO2 dissociates, releasing oxygen that enters into the tissues. And there is dissolved oxygen also that also enters into tissues. About 3%, about 97% of oxygen, which is carried to the tissues in the form of oxyhemoglobin, dissociates, releasing oxygen that goes to the tissues. And the Hb combines with hydrogen ions, followed by the dissociation of carbonic acid, HCO3 minus, HC plus. H plus is combined with hemoglobin. That's why the hemoglobin acts as buffer. Hemoglobin in RBC acts as buffer. Similarly, in plasma also, hydrogen ions are formed in bicarbonate ion. Hydrogen ions are combined with blood proteins. So, in the plasma, blood proteins act as buffer by taking up hydrogen ions which are produced by the dissociation of carbonic acid. Similarly, in the RBC, the carbonic acid dissociates by the enzymatic activity of carbonic anhydride into HCO3 minus H plus. HCO3 comes out from RBC and in reverse, chloride ions enter into the RBC. What is called chloride shift. To maintain ionic equilibrium, the chloride shift occurs at the tissues is what is called Hamburger's phenomenon. HAMBRGERS, Hamburger's phenomenon. That is, here, HbO2 dissociates to the Hb and O2. O2 is applied to the tissues. Hb combines with the hydrogen ions forming reduced hemoglobin or HHB. So, what are actually on now? What are the reactions which take place at alveoli? At alveoli. Alveolar surface. This is A. Now this is blood. RBC, plasma. Okay. Yet alveoli, oxygen enters into the blood. So oxygen from the air enters into the blood where that is transported in dissolved state. How much percentage? 3% of oxygen is transported by the plasma in dissolved state. And %97 of oxygen enters into RBC combined with hemoglobin forming HbO2 or oxyhemoglobin. Okay. Here at alveoli, PO2 is high. PO2 is high. At the tissues, PCO2 is high. So at high PO2, HbCO2, carbaminohemoglobin dissociates into Hb plus CO2. Hb plus CO2. That CO2 goes out into the alveoli. Hb is there. Hb is there. The hemoglobin, carbaminohemoglobin at alveoli dissociates because of high PO2. High PO2. Now carbon dioxide passes out. And another form, in the plasma, bicarbonate ions are there. Again they enter. Reverse chloride ions goes out. Reverse phenomena takes place at alveoli. So bicarbonates which are present in plasma now enter into RBC, and in reciprocation chloride ions goes out into the plasma. Now HCO3 minus. Here there is reduced hemoglobin, HHB, that dissociates into Hb plus H. That H combines with HCO3, forming H2CO3. That dissociates into H2O and CO2. CO2 passes out. So CO2 by the dissociation of carbonic acid and CO2 by the dissociation of carbaminohemoglobin. CO2 which is present in dissolved state in 3 routes, they enter into the air. Oxygen enter into blood and it enters into RBC. 97% of oxygen combines with the hemoglobin. Hemoglobin by that time it reaches the, it is in what state? It is in reduced hemoglobin state. So hydrogen dissociates and Hb combines with oxygen forming oxyhemoglobin. This is the reaction that takes place at the alveoli. Again, I am telling you, at the tissues, carbon dioxide is released from tissues, enter into RBC, and transported to the alveoli in 3 different forms. In dissolved state, 7%, in as bicarbonates, 70%, and as carbaminohemoglobin, 20 to 25%. Okay. And oxygen at the alveoli, is transported, is diffused into the blood and transported by the blood to the tissues in 2 forms. In dissolved state, 3%, in 97% as oxyhemoglobin. That is about the transportation of carbon dioxide. At tissues, carbon dioxide combines with water, forming H2CO3, dissociates into H and HCO3 minus. H combines with hemoglobin, forming reduced hemoglobin. This is what is called reduced hemoglobin. And bicarbonate ions, they move out. And chloride ions, they enter in. And the carbon dioxide is transported to the alveoli in the form of bicarbonate, 70%. At alveoli, bicarbonates again, they enter into RBC, chloride goes out. Bicarbonates and hydrogen ion combine to form H2CO3, dissociates into water and carbon dioxide goes out into the air. This reaction is characterized by carbonic acid. That is about the transportation of carbon dioxide. And another one, regulation of respiratory regime. 3 points are there here. Just you write down four important points. Regulation of respiratory rhythm. Respiratory rhythm, inspiration, expiration. Per minute, the rate of breathing is about 12 to 16 times. This is normal respiratory rhythm. So the occurrence of normal respiratory rhythm is under the control of neural system. First point, respiratory system regulation is under the control of the neural system. 1 minute, what did I do? I will continue. Just 10 minutes. So I want to close down respiratory system today itself. Regulation of respiratory system is controlled by neural system. Next to me. Normal respiratory rhythm is known as EUPLEA. EUPLEA. That means normal respiratory person having normal respiratory rhythm is what is called EUPLEA. EUPLEA. You write down this point. EUPLEA. Normal respiratory rhythm. And the respiratory rhythm is regulated by respiratory rhythm center. Respiratory rhythm center, important point, located in medulla of brain, medulla oblongata of brain. Primary respiratory rhythm is located in medulla oblongata of brain. This is the respiratory rhythm center. The regulation of the respiratory rhythm is regulated by respiratory rhythm center located in medulla. And another center, pneumotaxic center, PNEUMOTAXIC. Pneumotaxic center is located in pons of hindbrain. Pons varolai of hindbrain is a part of the cerebellum. There, the second respiratory center, regulatory center is located what is called pneumotaxic center. Signals from pneumotaxic center located in pons regulate the functioning of or moderate the function of respiratory rhythm center. The signals from pneumotaxic center can moderate the functioning of the respiratory rhythm center. You see, the inspiratory rate can be reduced by the signals generated from pneumotaxic center. By that, respiratory rhythm center can alter the respiratory rate. The inspiratory rate can be altered by the respiratory rhythm center on the signals received from pneumotaxic center. Next, third point. Chemosensitive area. Chemosensitive area is located near respiratory rhythm center. It recognizes the changes of the concentration of carbon dioxide and hydrogen ion. So the carbon dioxide concentration in the blood, the hydrogen ion concentration is in the blood, normally identified by another chemosensitive area located near the primary respiratory rhythm center. Chemosensitive areas, chemoreceptors, an area in which these receptors present they perceive the changes of carbon dioxide concentration and hydrogen concentration of the blood and make necessary and give necessary signals to rhythm center to find the solution. And another one. Chemoreceptors, which can perceive the changes of CO2 and hydrogen ion concentration. They can perceive the changes in the carbon dioxide concentration and hydrogen ion concentration in the blood. These chemoreceptors are present in the walls of carotid artery and aortic artery. So the walls of the carotid artery and aortic artery contain chemoreceptors which will recognize the changes of the CO2 and hydrogen ion concentrations. Oxygen has no significant role in the regulation of respiratory rhythms. Oxygen concentration in the blood plays no significant role in regulation of respiratory rhythm. Oxygen has no significant role in regulation of respiratory rhythm. This is another point. That is about the regulation of respiratory rhythm. Lastly, respiratory disorders. Asthma, characterized by wheezing sound, wheezing due to inflammation of bronchi and bronchioles. Wheezing and gurhala. When we respire, you see a rattling sound is produced in the air transport through the respiratory system. Wheezing sound. That is due to inflammation of bronchi and bronchioles. Asthma, difficulty in breathing. Next one. Emphysema. Damage of alveolar surfaces. Damage of alveolar membrane surfaces. Cigarette smoking is the main cause. Cigarette smoking is the main reason for emphysema. Coming to the third disorder, occupational respiratory disorders. Occupational respiratory disorders. These are found in the workers working in granite and cement factories. Due to inhalation of granite dust or cement dust for prolonged time, fibrosis takes place in the alveoli. Fibrosis, that means development of tissues. Fibrosis occurs in the alveoli that leads to the respiratory, occupational respiratory disorder. Sometimes it may cause cancer also. So this is about the disorders caused due to infections of the respiratory system. With this, respiration is completed. Tomorrow, we take up an important topic, physiology of excretion only, to be completed only in 2 hours. Physiology of excretion. Like I think most of the students can understand the physiology of respiration. We have a clear picture regarding the physiology of respiration. I explained in detail I took 5 classes to complete the respiratory system in the time without any doubt. If students fail to listen this online class, you see a video will be uploaded. Please see that video also. Please try to understand. Write down the notes while I'm teaching. Every student should write down the notes. Digital display of teaching is different from traditional type of teaching. In digital type, all notes are displayed on the notice board. Just what is the duty of a lecture? Just read out the sentences which are displayed on the digital display board. So for that, lecture is not required. Any man who knows English can read out the sentences displayed on the digital board. But that is not the correct approach. Lecturer should write the letters, write the sentences on the board, and he has to explain the subject. The subject should be come out from the mouth of the lecturer. Then only that is useful. I can change the sentences. I can formulate different types of questions externally. Whereas a distance display simply gives the information that is available in textbooks also. Or the digital display of teaching lecture is not required. Okay? With this, today class is completed. Tomorrow we will start another important physiological topic, that is the physiology of expression. Most important. I don't want to go into the structure of the excretory system, just physiology of expression. Very good. Thank you very much. All students are requested to go through the video recordings also. For the convenience of students, most of the students express that it is not possible for us since we are otherwise engaged between 10 and 11 a.m. So, in view of the inconvenience of the students, I am uploading the video classes also. So, whenever you find time, you please look into the videos. Okay? Very good.